سڵاوکردگانی بەڕێز من دکتۆر سادات بوو بۆ هەندێک لە وانەکانی فیزیۆلۆژی لەگەڵتانە بن ئەمڕۆ باسی blood physiology ئەکەین. So today our body tissues made of different types of tissues, including the epithelial tissue, muscle tissue, nerve tissue, and connective tissue. Blood is a type of the connective tissue. We have different types of connective tissue based on the structure and the components of the tissue. In the case of the blood, what we have different types of the cells, which are suspended in a watery matrix or tissue matrix, which is called the plasma. So blood makes up about 8% of the total body weight. If you drain the whole blood from the body of a person, we are expecting to lose about 8% of its body weight. So the blood is made of two main compartments. The first compartment is the blood cells, if you have a look here, and the cell fragments. About 38 to 48% of the blood composes of various cell types, including the erythrocytes, which have the, let us say, their own specific shape, and then we have different types of leukocytes. We will explain them later in detail. So these are all the leukocytes, and what we have the platelets or thrombocytes. Thrombocytes, they have their own characteristic features. So, and they are considered as cell fragment. Why? Because the megakaryocyte, which is the progenitor of these cells, cannot penetrate the bone marrow capillary bed, and they undergo a process which is called the cell defragmentation, and they send their fragments into the circulatory system. So we don't have an intact megakaryocyte within the circulatory system. So that's why the platelets or thrombocytes are recognized as the cell fragment. So the remaining portion of the blood is made of the plasma. This watery background is the plasma, and it is about 52 to 62% of the blood is made of the plasma. It is just it can act as a watery background, and the cell and the cell fragments are suspended in this. Similar to that of the other tissues, the blood has some properties. The first one is the amount of the blood. As so mentioned earlier, about 8% of the blood is of the body weight is made of the blood. Normally, female has about 4 to 5 liters of blood, and male has a bit higher amount in compared to female. It has about 5 to 6 liters of the blood. So the second property of the blood is the color. Normally, the arterial blood has a brighter color in compared to the venous blood. Why? Because the arterial blood is normally saturated with oxygen. I think you might have an idea about this, while the venous blood is normally occupied with what? With the carbon dioxide and waste products. So that makes the venous blood a bit darker in compared to what? To the arterial one. And the third property of the blood is the pH. It's the pH of the blood. So blood pH is ranged between 7.35 to 7.45. So venous blood normally has a lower pH than does arterial blood because of the presence of a high amount of carbon dioxide, as we mentioned earlier. So when the carbon dioxide within the blood is form a complex with what? With the H2O. So H2O, when it is reacted with the water, it forms a complex which is called H2 CO3. So it is this complex is not let us say is not a stable complex. So it is dissociated to form hydrogen and HCO3. So when you have hydrogen within a medium or within a solution, it makes it a bit acidic in compared to what? To a solution without the hydrogen. So that's why the venous blood has a bit lower pH in compared to what? To the arterial one. And the last property of the blood is the blood viscosity. So viscosity means thickness or resistance to flow. So as you know, blood has a colloid texture, and this texture make it a bit thicker in compared to water. So normally, blood is about 3 to 5 times thicker than water. Generally, blood has different functions. The first one is the transport of gases, nutrient, and waste products. So the main function of the blood is the transportation of the gases, nutrient, and waste products. As you know, from the respiratory system, the blood can carry oxygen to the body tissues and returning the carbon dioxide from the tissues back into the respiratory system and the other systems to be eliminated from the body. And also, it is responsible for the transportation of, let us say, different types of nutrients or dietary or required diets from where? From the or materials from where? From the gastrointestinal tract, from the digestive system into the different types of tissues to be used as a source of energy and for building up the tissues. And from the tissues, it can return back the formed waste products into the different tissues, including the kidneys, liver, and the other organs to be eliminated. Then the second property is the transport of the processed and regulatory molecules. So as you know, some of the molecules in our body has a specific function, such as the hormones and some types of the regulatory hormones molecules and processed molecules such as the precursor of, let us say, vitamins, including the vitamin uh, D, can be transported from the circulatory system to the target or to the tissue cells. And then blood also can regulate the pH and the osmosis of the body. As we mentioned earlier, the blood has its own specific pH. So the pH of the tissues, as you know, um, can be altered as a result of metabolic activity of the tissues. So how the changes in the pH can be regulated, so with the aid of the blood. And then, as you know, normally our body has a specific, let us say, osmolarity or osmosis. Uh, so, with the aid of the blood, we will explain this later. So, with the aid of the blood, the process of osmosis can be regulated. So, as an overview, osmosis or let us say osmolarity or osmosis is the movement of water from a more watery side to a less watery side. So the pH and the osmosis of the body can be regulated through the blood. Another function of the blood is the maintenance of the body temperature. As you know, our body has let us say, a specific temperature, which is ranging between 36.5 to 37.5. And the core body temperature can be regulated through the core body temperature can be maintained through the blood. And then also blood has a property which can protect our body against foreign substances. If you remember what we said before, we said within the blood, what we have different types of cells. One type of the cell is the white blood cells. The white blood cells are responsible for fighting against the especially the microbial agents, and they can protect our body against different microbes. Also, within the blood, we have some specific molecules which are recognized as the antibodies. So also the antibodies can be used as a defense mechanism against the microbes. Then the other property of the blood is the maintenance of the vascular system. When there is an injury or when there is a damage in the wall of the blood vessels, so it can be repaired with the aid of some specific cell fragments which are recognized or called as the platelets in a process which is called hemostasis. So hemostasis is the prevention of the blood loss. So it made of different stages. We will explain it later. So blood can maintain the vascular system or repair the any damages in the vascular system. So now I'm trying to explain the plasma. So plasma is a liquid part, if you remember, what we said the cell and cell fragment is suspended in a tissue matrix. The matrix was we is called the plasma. So plasma is the liquid part of the blood, which is a pale yellow colloidal fluid in which different cell types are suspended. So normally the uh, plasma has a pale yellow color. So any changes in the plasma color might indicate a specific condition. So if you have time, you will explain this in detail. And the plasma has a colloidal texture. Why? Because it made of different compartments. Normally, about 98% or 92%, sorry, of the plasma is made of what of water, and about 7% of the plasma is made of what proteins, which including the albumin, globulin, and fibrinogen. So the albumin has different functions in our body. The first one is the mainly it is involved with the production of a specific pressure, which is known as plasma uh, colloid osmotic pressure, and this pressure is responsible for the maintenance of a balance between the intra and extravascular fluid flow. Okay. Also, albumin has, let us say, other functions. It can act as a carrier proteins within the circulatory system. And what we have the globulins. The globulins also they have different roles. Mainly they are involved with the immune system. And fibrinogen it can act as a or it has a role in the process of hemostasis. We will explain it later. The majority of plasma proteins, not all, the majority of the plasma proteins are produced by the liver. So any defect in the liver might have a negative impact on what on the level of the plasma proteins. So any defect in the liver might have a negative impact on what on the level of the plasma proteins, and any changes in the plasma proteins has a negative impact on what on the plasma colloid osmotic pressure. We will explain this pressure later. So the remaining portion of the plasma, it is about 1% of the plasma, is made of the electrolytes, including the sodium, potassium, or the other types of electrolytes, waste products, which is um, uh, produced or as a result of metabolic activity of the tissue cells, nutrients which can be absorbed from the GIT and transported through the blood, and the other substances. So plasma minus fibrinogen and clotting factors is called serum. So serum is a plasma without fibrinogen and the clotting factors. Uh, so plasma uh, has some specific functions, or it is involved with the regulation of some processes. The first one, the uh, plasma proteins, uh, they can act as carrier proteins, so they can transport different molecules throughout the blood. For instance, when you have hydrophobic molecule in the circulatory system, if it is a hormone or other agent is, so it can be carried or transported through the plasma through the blood by binding to, let us say, specific proteins, including the albumin and the globulins, and they can be transported through the circulatory system. And the plasma proteins, they can act as the, let us say, defense mechanism. Why? Because if you remember, when we explained the plasma protein, we say globulins mainly involved with the defense mechanism, because we have, let us say, different antibodies, types of antibodies in our circulatory system, and these or those antibodies can act as a line of immune system, and they can defend our body against different types of microorganisms. Plasma proteins are a good buffering system of hydrogen ions. So when there is an accumulation of carbon dioxide within the circulatory system, the level of hydrogen ion might increasing, and because of the presence of the proteins in the plasma, it can neutralize the high level of hydrogen, and so it can act as a buffering system. Also, some of the plasma proteins can act or they can maintain the vascular system in a process which is called coagulation or hemostasis, and fibrinolysis. So hemostasis, as we mentioned earlier, is the prevention of the blood loss. So we will explain this process in detail. So other process which is related to the hemostasis is the fibrinolysis. After repairing of damaged blood vessels, so the remnant of the clotting process can be removed in a process which is called the fibrinolysis. Also, this process is active in a person who has a stroke or other type of infarction. Also, the plasma proteins can act as enzymes. We have different types of enzymes in our circulatory system or in our blood. So they can, or molecules or proteins, they can analyze a specific type of chemical reactions. So another function of the plasma proteins is the maintenance of the plasma colloid osmotic pressure. I think already we mentioned a bit about this pressure. So the colloidal texture of the blood plasma is due to what? Due to the presence of plasma proteins, and contributes in the creation of a specific osmotic pressure, which is called the plasma colloid osmotic pressure. So the plasma colloid osmotic pressure is responsible for what? For the controlling of a balance between what? Between extra and intravascular fluid flow. So what we mean by intra and extra vascular fluid flow? It means when we have, like, let us say, a persistent fluid flow between what? Between the blood and the extravascular tissues. So how a balance between these fluid flow can be regulated through the presence of a specific pressure, which is called plasma colloid osmotic pressure, and this pressure is mainly depending on what? On the presence of the plasma protein. So any change in the level of the plasma proteins might lead to imbalance between the intravascular and extravascular fluid flow. So, as you know, the fluid can move from a site to another site through a process which is called osmosis, and it is a diffusion of water through selectively permeable membrane from a more watery to the less watery side. So from a side where the solutes, we have let us say low concentration of solutes to the side where you have a high concentration of solutes. So the force that stop the process of osmosis is called what is called the osmotic pressure. So this force, which prevent the movement of the water from let us say a more watery side to the less watery side, is called what is called the osmotic pressure. So this pressure prevent the further movement of the water from the site where you have more water into the site where the amount of water is lower in compared to what to the other side. So any changes in the level of the plasma proteins might lead to what? To a condition which is called hypoproteinemia. So what is the hypoproteinemia? Hypoproteinemia is a reduction in the total plasma protein concentration than the normal range. If you remember, when we explained the plasma, we said about 7% of the plasma is made of plasma proteins. So and what we said the majority of these proteins are made by the liver. So any defect or any factors which has a negative impact on the protein levels in the blood might lead to what? Lead to the hypoproteinemia. So reduction in the level of the plasma or plasma proteins. So when there is a reduction in the plasma proteins, this lead to what? Lead to a specific condition or changes in the plasma colloid osmotic pressure, the pressure which is responsible for the controlling of the extra and intravascular fluid flow. So when there is, let us say, imbalance in the fluid flow between the intravascular and extravascular tissues, this lead to what? Lead to a condition which is called edema or edema, which is characterized by the accumulation of what? Of body fluid in the extravascular tissue and body cavities. Accumulation of fluids in the extravascular tissue and the body cavities. So as you know, we have different types of cavities in our body, including the thoracic cavity and the abdominal cavity. And this might be resulted from different causes or factors. The first one is the starvation. So during famine or during other, let us say, other or factors or other, let us say, or when there is a reduction in the resources of protein, so this might lead to what? Lead to the reduction in the level of the plasma proteins, and this might end up with what? With the edema. The second main cause of the edema is the disturbed protein synthesis by the liver. If you remember what we said about um, or the majority of the plasma proteins are made by the liver. So when there is any defect in the liver as a result of the liver damage or uh, liver defects, might lead to what? Lead to the edema. Any defect in the liver as a result of liver damage uh, might be related to the liver cirrhosis, liver cancer, viral infections of the liver, and we have, let us say, different types of factors which might affect the function of the liver. And the third um, cause of the edema is an increase in the protein loss, either through the gastrointestinal tract, which is called the protein losing enteropathy. So it means losing the protein as a result of intestinal diseases. Pathy, it means what? Diseased intestine. Or through the kidneys, which is called protein losing nephropathy. So it means the or the body or the plasma proteins might be lost through where? Through the kidneys as a result of the renal damage. So we are done with the plasma, and we mentioned the most structural components of the plasma and the role roles of the plasma in the circulatory system. So now I'm trying to explain the cells of the blood. So generally, we have three main types of the blood cells, and they have, let us say, a different morphology and functions. The first and the most dominant type of the blood cells uh, is called the red blood cell or erythrocyte. Okay, and the second type of the blood cells is the white blood cells. We have different types of white blood cells. We will explain this later in detail. So each of these cells has its own uh, function in the circulatory system. And the third type of the blood cell or cell fragments is the platelet or thrombocytes. Okay. So origin and sites of the blood cell formation. So blood cells are produced from a specific type of cell, which is called the pluripotent hematopoietic stem cells. Pluri, it means it can differentiate into different types of cells. Potent, so it means they have a potency to differentiate and form different type of the cell. Hemato, it means related to the blood. Hematopoietic, it means blood-forming cells. Stem cells, the stem cells are undifferentiated cells, so the stem cells are the progenitor of our body cells. So normally the blood cells are produced from this type of a cell, which is called the pluripotent hematopoietic stem cells. The pluripotent hematopoietic stem cells have surface uh, receptors for specific stimulatory uh, messengers, or let us say hormones or signals, including the erythropoietin. Erythro, it means the red blood cells. So erythropoietin is responsible for the induction of the pluripotent hematopoietic hematopoietic stem cells to differentiate into what? Into erythrocytes. When the erythropoietin bind to the, let us say, its own specific receptors on the pluripotent hematopoietic cells, the pluripotent hematopoietic stem cells directly start uh, or they directly differentiate into what? Into erythrocytes. Also, this hormone or this, let us say, ligand is called the thrombopoietin. Thrombo, it means what? It means the thrombocytes or platelets. Thrombopoietin is responsible for the induction of the pluripotent hematopoietic stem cells to differentiate into what? Into thrombocytes or megakaryocytes. Okay, so when the thrombopoietin binds to its own specific receptors on the surface of what? Of the pluripotent hematopoietic stem cells, it this these cells are directly start differentiate differentiation, and it form what? It form the uh, uh, thrombocytes. Then we have another hormone, which is called the colony stimulating factor. So this signal or this message is responsible for what? For the differentiation of the pluripotent hematopoietic stem cells into different types of the leukocytes. So in the early weeks of embryonic life, the primitive, uh, it means the primary uh, cells or blood cells, which are nucleated blood cells, are produced in the yolk sac. Yolk sac is a specific structure which is related to the process of what? Of the embryogenesis. So the uh, primary or nucleated blood cells, red blood cells are normally enucleated cells, so they don't have nucleus. So in the early weeks of embryonic life, the circulated red blood cells and the other types of the cells are nucleated, and they are originated from where? Produced by the yolk sac. During the middle trimester of gestation, as you know, we have in the case of human, there is three, let us say, uh, trimester of gestation. So in the middle trimester of gestation, the liver became the main site uh, for the process of hematopoiesis. So the liver is responsible for the production of the blood cells. And during the last month of gestation and after birth, the blood cells are exclusively produced by what? By the bone marrow, which is in a process which is called the medullary hematopoiesis. So the medulla, the bone marrow, which is located within the medulla of the bone, became the main site of the process of hematopoiesis. So the production of the red, or so let us say the blood cells, including the red blood cells, white blood cells, and platelets. So what is the red blood cell, or what are the red blood cells? The red blood cells are enucleated. It means they don't have nucleus, okay? Biconcave disc-shaped cells. So it means they are compressed from both sides, and they have, let us say, a biconcave disc-shaped cells, and the diameter of the red blood cells is about 7.5 micrometer, with the, let us say, a thickness of 2 micrometers, with the thickness of 2 micrometers. And the red blood cells are packed with what? With a specific type of protein, which is called the hemoglobin. We will explain this later. So the red blood cells or the red blood cells can act as a sac of hemoglobin. Normally, there is about 5 to 5.5 million cells per microliter, or uh, 5 to 5.5 million uh, erythrocytes in the, let us say, a microliter of blood in male, and there is about 4.2 to 5.4 million cells per microliter of blood in female. So normally male has a higher number of the circulated erythrocytes in compared to what? To female, and this is related to some, let us say, variations or some factors are related to what? To the genders. So what is the advantage of having a biconcave disc-shaped cells in the case of red blood cells? So um, this feature increases the surface area of the red blood cells. So the greater surface area makes the movement of the gases into and out of these red blood cells more rapid and efficiently. So the gases, especially the oxygen, can easily enter into the cell and combine with what? With the hemoglobin, and it can leave it when it gets into the body tissues where the partial pressure of the oxygen is a bit lower in compared to the um, erythrocytes. So the first advantage of having such a kind of a shape, biconcave disc shape, is to increase the surface area of the cells. And the second, let us say, advantage of having such a kind of shape is to enable the red blood cells to bend or fold around its thin center. So the red blood cells has a thinner center in compared to the periphery or the edge of the cells. So they can easily bend and fold when they are passing through let us say, a blood vessel or a capillary which has a smaller diameter than the red blood cells. So if during the process of erythrocyte aging, the red blood cells might lose its flexibility, and when they are passing through a blood vessel or a capillary which has a smaller diameter than the red blood cells, this lead to what? Lead to the hemolysis or rupturing of the red blood cells. So having a biconcave disc shape of the erythrocytes has two main advantages. The first one is the increase in the surface area of the cell, and the second one is to enable the cells to bend and fold around its thin center when they are passing through a blood vessel or a capillary which has a smaller diameter than the cells themselves. So the blood cells or the RBC synthesis can be regulated through a process which is called erythropoiesis. So normally when there is a reduction in the level of oxygen in the circulatory system or at the tissue level, which is called the hypoxemia, when there is a deficiency of the oxygen in the blood. So these changes can be detected by specific cells which are located within the kidneys, and they produce what they produce hormone. I think we already mentioned this, which is called the erythropoietin. So any reduction such as having anemia, which lead to what? Lead to the reduction in the level of oxygen in the blood. High altitude for some those people who are living at the places which is a bit or let us say the partial pressure of the oxygen is a bit lower in compared to the sea level, such as the Tibetan people. Heart diseases, again, because of the, let us say, defect in the pumping of the blood to the respiratory system, this might lead to what? Lead to the reduction in the level of the oxygen in the body. And we have, let us say, other factors, such as respiratory defects, fibrosis, or the other factors of respiratory system might lead to what? Lead to the reduction in the level of the oxygen in the circulatory system. And this lead to what? Lead to a condition which is called hypoxemia, which is a deficiency in the level of the oxygen in the circulatory system. And this change can be detected by what? By some specific cells within the kidneys, and they release what? They release a specific factor which is called the erythropoietin. So they produce what? They produce a specific hormone which is called erythropoietin, and the erythropoietin enter into the circulatory system, and through the circulatory system they can get to where? To the bone marrow. So they induce what? They induce, they bind to a specific receptors on the surface of the pluripotent hematopoietic stem cells, as we mentioned earlier, and they induce them to produce what? To differentiate into what? Into erythrocytes, and they became what? Pro-erythroblasts, and the pro-erythroblasts, they can differentiate further to become a mature red blood cells. And when you have, let us say, a sufficient number and amount of the red blood cells in the circulatory system, you are expecting, let us say, an increase in the level what? Or returning the oxygen partial pressure back into its normal level, and this process can be stopped or can be switched off when there is let us say a sufficient amount of the oxygen in the circulatory system. So the process of erythropoiesis can be regulated by what? Or can be affected by what? By any changes in the level of the oxygen within the blood. Okay. So how the red blood cells can differentiate into a mature erythrocyte. So the process of erythrocyte differentiation is a complex process, and I try to abbreviate like this. So what we have, as we mentioned earlier, so we said the process, the progenitor of the erythrocytes are the pluripotent hematopoietic, or the blood cells are the pluripotent hematopoietic stem cells. These cells have, let us say, specific receptors for the hormones, which are responsible for the blood cell production. So the pluripotent hematopoietic cells are partially committed hematopoietic cells. Why they are partially committed hematopoietic stem cells? Because they can differentiate only into what? Into the blood cells, different types of blood cells. So the pluripotent hematopoietic stem cell can produce white blood cell, megakaryocyte, and red blood cells. So they cannot differentiate into muscle or, let us say, connective tissue and nerve tissue. That's why they are partially committed to become what? To become only the blood cells. So they can be induced through with the aid of the erythropoietin into what? Into pro-erythroblasts. So the pro-erythroblasts are committed hematopoietic cells that can be differentiated to erythrocytes only. So what you have here, this is a committee. So they can only differentiate into what? Into erythrocytes. But in the case of pluripotent hematopoietic cells, we said partially committed. Why partially committed? So they can only form the blood cells, including the white blood cell, platelet, and red blood cells. But they cannot produce other types of the body tissues. While the pro-erythroblasts are committed hematopoietic cells, meaning they can only differentiate into what? Into erythrocytes or the red blood cells. And then these cells can further differentiate into what? Into reticulocytes. The reticulocytes are immature red blood cells. Normally, the percentage of the reticulocyte is slightly less than 0.1% of the circulated red blood cells, and it contains, why they are called reticulocytes, because the cytoplasm of the cells contains, let us say, a small amount of remnants of the Golgi apparatus, a bit of mitochondria, and a few other cytoplasmic organules, which are not normally present in a mature red blood cell. So mature red blood cells has no mitochondria, has no, let us say, remnants of Golgi apparatus and other cytoplasmic organules, but the reticulocyte still has a bit of them. So they can, the reticulocytes can circulate for about 1 to 2 days. Then they differentiate into what? Into a developed mature red blood cell. So an increase in the number of reticulocyte, or which is called reticulocytosis, can be seen in some cases of anemia, especially the regenerative anemia. So we have two main types of anemia. The first one is called non-regenerative anemia, and the second one is the regenerative anemia. The non-regenerative anemia is mainly related to the defects within the bone marrow, while the regenerative anemia might be related to the other factors such as iron deficiency, folate and B12 deficiency, and the other factors. So when there is an increase in the number of the circulatory reticulocytes, either due to, let us say, treatment of the anemia or the other factors, it means the anemia is regenerative, so it is a treatable condition. If you remember what we said the red blood cells can act as a sac of hemoglobin. So they are occupied with a specific type of protein, which is called hemoglobin. So hemoglobin is a globular molecule that made up of 4 subunits. So globular, it means they made up of a specific type of protein which has a globular shape, and each molecule of hemoglobin is made of 4 subunits. If you have a look here, what you have, this is a structure of hemoglobin: 1, 2, 3, 4. So it's made of what? All of 4 subunits. Each subunit contains a heme, this blue painted spots are hemoglobins. So each of the subunits contains a heme prothetic group, the functional group of the hemoglobin, which composite of what of this is if we enlarge one of these structures, it became something like this. This is the structure of hemoglobin. As we mentioned earlier, it's made of 4 subunits: 1, 2, 3, 4. So uh, the each of these subunits is made of what? Of a heme prothetic group, which contain a, this is a structure of a heme, okay? This, which is contains an iron atom, which is located at the center of a structure which is called the protoporphyrin ring. This is the protoporphyrin ring, and what we have an iron atom within the center of the protoporphyrin ring. Okay, so this is a structure of what? Of a heme, which is the functional unit of the red blood cells. So heme is made of what? Of an iron atom and a protoporphyrin ring. So the heme is conjugated, as we mentioned before, with a polypeptide. The polypeptide, this structure, the folded structure, is called the hemoglobin. So the heme, the prophetic group of the hemoglobin, is conjugated or combined with what? With a globular protein, which is called the globin. So they form a hemoglobin subunit. So what we have a heme and a globin. They form what? They form a hemoglobin subunit. So when 4 hemoglobin subunits combine, they form what? They form a complete structure of the hemoglobin. So each red blood cell contains approximately about 300 million hemoglobin. So each red blood cell is occupied by around 3 million hemoglobin molecule, each of which can carry 4 oxygen molecule. Imagine the amount of the oxygen which can be transported through the through a single red blood cell. So what we have let us say around 5 million red blood cells in our per microliter of blood. So each red blood cell can carry about so contains about 3 million hemoglobin, and each hemoglobin can carry about 4 oxygen molecules. So imagine the amount of the oxygen which can be transported through the blood. The hemoglobin can be produced from different precursors, which include the succinyl-CoA and glycine. So the succinyl-CoA and initially the succinyl-CoA and glycine are combined in a chemical reaction to form a pyrrole ring. So this is a pyrrole ring. This is a pyrrole ring. If you have a look here, and this one is also a pyrrole ring. What we have 4 pyrrole ring combined to form a protoporphyrin ring. So 1, 2, 3, 4. So at first, initially, as I said, succinyl-CoA and glycine combine to form what? To form a pyrrole ring. Then 4 pyrrole ring combine to form what? To form a protoporphyrin ring. 1, 2, 3, 4. They form what? They form a protoporphyrin ring. Then the protoporphyrin ring and an iron atom, this one, the protoporphyrin ring, the whole ring, with the an iron atom, they form what? They form heme, which is the prophetic group of the hemoglobin. Four heme and four globin molecule combine to form a complete structure of hemoglobin. So hemoglobin molecule is made of what, as we mentioned earlier, is made of four hemoglobin subunits. Okay, four hemoglobin subunits. Okay. The primary function of the red blood cells is gas transport. So the red blood cells are mainly involved with the gas transport between the respiratory system and the tissue cells through the cardiovascular system. So the first gas which can or mainly or uh, the which is mainly transported by the blood is the oxygen. So approximately about 98.5% of the required oxygen can be by the body can be transported through the in combination with what, with the hemoglobin. So in a rapid and reversible manner. So what we meant by rapid, so it means the oxygen, when there is a reduction in the partial pressure of the oxygen at the hemoglobin level, the oxygen directly combine with what, with the hemoglobin to form what, to form oxyhemoglobin. And when it is a reversible process. Why it is a reversible? Because when the red blood cells get to the tissue level, where there is the partial pressure of the oxygen is lower in compared to the respiratory system, so the oxygen can directly leave the hemoglobin structure. So it is a reversible process, and they can enter into the tissue and they can be used to form energy. So as we mentioned earlier, about 98.5% of the oxygen should be transported through the hemoglobin in the form of oxyhemoglobin. About 1.5% of the hemoglobin can be transported as a dissolved form in plasma. So it is only, let us say, a small amount of oxygen can be transported through the plasma, while the vast majority of the oxygen should be transported in the form of oxyhemoglobin. While the carbon dioxide is, let us say, partially transported through the hemoglobin, because approximately about 7% of the carbon dioxide is dissolved in plasma, in the plasma of the blood. About 23% of carbon dioxide is transported in combination too with the hemoglobin. Look, if you remember what we said about 98.5% of the oxygen should be transported in the form of oxyhemoglobin, while only about 23% of the carbon dioxide can be transported through the hemoglobin in the form of carbaminohemoglobin. So the amount of the transported carbon dioxide through the hemoglobin is about fourfold lower in compared to the oxygen. Normally, about 70% of the carbon dioxide can be transported or as a dissolved form within the cytoplasm of the red blood cells. So what we have, if you have a look at this diagram, when the CO2 get into the red blood cells, they can either transport it through the hemoglobin. It is only about 23% of CO2, and about 70% can be dissolved within the cytoplasm of the red blood cells. So the dissociation, or the let us say the process of dissolving of carbon dioxide within the cytoplasm of the red blood cells is facilitated through the presence of a specific enzyme, which is called carbonic anhydrase. This enzyme is facilitating the combination between what? Between carbon dioxide and H2O to form what? To form a, let us say, less stable molecule, which is called H2CO3. And the H2CO3 is directly dissociated to form what? Hydrogen ion and bicarbonate, HCO3. So then when it gets to the respiratory system, or when it gets into the kidneys, they can be eliminated as HCO3, or it became what? At the level of the respiratory system, they can be eliminated. So the carbonic, or let us say the any defect in the function of carbonic anhydrase might interfere the process of carbon dioxide transportation.